Alright, so last time we're talking about maturation of red blood cells and the significance of folic acid and vitamin B12. In this video, we're going to talk about erythrocyte death and disposal, iron metabolism, and jaundice. Right. So this is the basic life cycle of red blood cells. You see that red blood cells have a lifespan of 120 days, after which they become old and have to be removed, right? As they will take up unnecessary space and they will no longer be as efficient in carrying oxygen throughout the body, right? They are now said to be expired erythrocytes, as they have lost their flexibility, resilience, and durability, right? So we see that in the life cycle of red blood cells, they have to be uh, continuously broken up and replenished. And you see that they are broken up in the liver and in the spleen, and the byproducts are then used. They are recycled again to make more red blood cells. And we also see that instead of breaking them down, uh, the cycle can actually even continue from the GIT, where we absorb nutrients such as amino acids, iron, and folic acid to make these red blood cells via erythropoiesis in the bone marrow, right? So we see that the disposal of these red blood cells occurs mostly in the spleen, right? So this is the spleen. It consists of two pulps. We have a red pulp and a white pulp, and the red pulp acts as a filter and a checkpoint for red blood cells. It contains a tissue macrophage system which breaks down old or senescent red blood cells, right? And we see that this spleen contains a narrow openings, right? As I said, which act as a checkpoint or a filter or a distinguisher, or it acts to stop the passage of old red blood cells. And we see that since these sinuses have a small narrow opening of 3 micrometers, when old red blood cells, which have a diameter of 7.5 micrometers, try to pass through these sinuses to re-enter circulation, right? They get trapped, right? They get trapped as they are no longer able to easily squeeze through these sinuses, right? And when they get trapped, they get attached by a binding protein called laminin A5, and this laminin A5 attaches to this old red blood cell. And we see that due to the position of the old red blood cell in a small space of 3 micrometers, it kind of hemolyzes, right? It breaks down, it bursts into 3 components. We have this plasma membrane component, and we have the globin component and the heme component. And the plasma component is just easily broken down by uh, red pulp macrophages. Firstly, the macrophage identifies this old red blood cell by the laminin A5, and it engulfs it simply, Then the globin part, right, it attaches again to a receptor on the macrophage, and it is broken down into amino acids, right, which are then used as normal amino acids from the diet. But this heme part now is a bit tricky, because firstly, the macrophages have to remove iron from this heme and release it into the blood, where it combines with transferrin. We shall talk more about transferrin in the next upcoming slides. And the macrophages then convert the rest of this heme into a greenish pigment called biliverdin. Then this biliverdin is further converted into a yellow pigment called bilirubin, right? And this bilirubin is then transported into the blood, and it combines with albumin. It's transported with albumin, right, in the blood to the liver, where in the liver there's a separation between albumin and bilirubin, and this bilirubin can be either stored directly into the gallbladder, which is the storage site of bile, or it can just be excreted as bile, right? And we see that the gallbladder discharges this bile, right, into the small intestines, where bacteria convert this bilirubin into urobilinogen, right, into urobilinogen, which is excreted as feces, or it can be in another pigment of urochrome, which is excreted or discharged as urine, right? Now, iron, it is an important it's an important element used for the formation of hemoglobin, myoglobin, cytochromes, and peroxidases. And its total body mass is around 4 to 6 grams, and 65% of it is found in hemoglobin, and 1% in other heme compounds, and 4% in myoglobin, and 15 to 30% stored principally as ferritin, right? So ion transport and metabolism, right? We see that whenever we have free iron, right, let's say from our diet or from the breakdown of this old red blood cells, right, or the degrading of hemoglobin, this free iron, when it's in blood, right, it gets attached to a transporter protein called transferrin, and this transferrin is loosely bound to this iron, and it then gets transported to multiple tissues as this iron transferrin complex passes through the blood. We also see that free iron, as we said in the last slide, is principally stored as ferritin in the liver, that is the storage organ, and it's stored in small quantities as hemosiderin, right? And this free iron can also be used to make heme, and also this free iron, now when it's in the form of this complex, the transferrin complex, can be excreted in feces daily, that is 0.6 milligrams, and it can also be used again to make new red blood cells, and it can be lost in menaces as 0.7 milligrams, right? So this is just the basic overview of iron transportation and metabolism. We break down iron, or we ingest iron, and we transport it as transferrin, and we store it in the liver as ferritin and hemosiderin, in high quantities as ferritin, and in small quantities as hemosiderin, and some of it is transported, is converted to heme, and other enzymes are broken down into free iron. So this is just a simple overview. Now, jaundice, right? It's a condition that arises when free or conjugated bilirubin accumulates in the blood. That is, free bilirubin ions, right? It causes the skin and the mucous membranes to turn yellow, as you can see in this diagram, and it arises when the total plasma bilirubin is greater than 2 milligrams per deciliter, right? So the reasons of the high levels of bilirubin, we see that excess production of bilirubin, for example, when one has hemolytic anemia, where red blood cells just start bursting, or they are broken down in high quantities by the spleen. This can lead to excess production of bilirubin, and also decreased uptake of bilirubin in hepatic cells, that is, in cells of the liver. This can also lead to the jaundice, as the concentration of bilirubin in the blood will be high. Also, disturbed intracellular protein binding or conjugation, in which there is failure of this bilirubin to be conjugated or to be processed by the liver into bile, right? And also there is disturbed secretion of conjugated bilirubin into the bile canaliculi, that is, into the bile ducts, right? There is failure of transportation of this conjugated bilirubin, that is, bilirubin which has been processed, into the bile duct and into excretion. Another condition is bile duct obstruction, where in the bile ducts there's maybe stones which kind of stop or impede the flow of bile into the small intestines and ultimately into its secretion, right? It can also be another cause of jaundice, right? Yeah, so that's all. Thank you for watching, and in the next video we're going to talk about anemia and polycythemia. So thank you.